I was originally scheduled to be your luncheon speaker, and I had thought I would be following a whole series of people who had talked about the wonders of the microbiome that lives on us. Instead, I'm coming at the beginning. And so my talk may seem slightly out of the order of what's going to be going on today, but given that this is a program on entrepreneurship, I have to say that this is an area that is ripe for study. Okay, how does this work? This statistic gets thrown around a lot. There was a study published in 2001 that said that modern people are inside 90% of the time. Actually, many of us, when we look at our own lives, might say, if you count being in the car, being inside, it's more like 98% of the time. And as you probably know, there is increasing worry that the places that we live inside might not be as healthy as a lot of aspects of so-called Mother Nature. There are terms that are very difficult to define that get bandied around a lot. Building-related illness and sick building syndrome. They're frequently used interchangeably. Purists separate them and say that building-related illness is when we know what's making you sick, something like Legionnaires. Sick building syndrome is when you get sick and you think it has to do with being inside someplace, your school, your office, your home, and you don't know why you're sick. And so a great deal of the impetus for doing microbiome research on buildings of all kinds is to see if there's something about the microbiome living in the physical structure that might be associated with these many known and unknown potential health risks. And right there we run into trouble. I'm chairing a National Academy study right now on microbiomes of built environments, and every meeting we just get sucked into how many different kinds of structures there are. And so recognize that, like all microbiome research, we're in at the beginning, and there's a great deal we don't know. And because buildings and it's of all types, structures, we're ranging from the space station to Neolithic caves and a lot of stuff in between, buildings like this, old-fashioned buildings, on and on. And so it's very hard to make generalities. I'm going to give you some background. Then I'm going to tell you my own personal story, how I got involved in it, and then a quick summary of some of what we know in very broad strokes. The World Health Organization and the Institutes of Medicine have done several meta-analyses trying to find associations between microbes and human health indoors. And we know without a doubt that increased allergies, increased allergic asthma, increased respiratory symptoms. We also know that frequently the indoors can be termed a microbial desert. It's not as great a place for a microbe to live as the soil or pond or the human gut or even the human epidermis. That said, we have been studying it for a long time, and a lot of the original research on microbes inside was done with the intent of seeing that they don't rot our stuff under the heading of biodeterioration. And I'm not going to go there today, but we have a century of work plus on biodeterioration. As we've gotten better at it, we did all the microbiology things. We'd go in and we'd use pieces of tape, or you can put out petri dishes in a room and see what settles. We've also invented various ways of sucking the microbes in that's particularly good at picking up spores and fungal spores, and there are lots of pieces of equipment that have been developed and attempts to standardize them. having said that the indoors is kind of a microbial desert, there's a big caveat. You get it wet, and all those microbes that have been lurking around are going to think that it's time to start growing. And we've all seen the gross things. It's frequently fungus that, you probably have some of this in your own house. It's very common in a basement around the washing machine to see something growing on the wall, combination of mold and gunk, and we haven't even begun to look at the viruses there. As a person with roots in mycology, fungal genetics, I've collected some pictures like that, and I've been in homes that look beautiful, where there will be a patch of mushrooms growing up in the corner of the basement. I mean, it can be pretty funny. There's a moisture problem when you see that. It doesn't take any microbiome research. So we know that when things get wet, that's when we have problems. We know that lots of people in the United States have allergies and asthma, many of which can be associated with mold spores and other microbial things. And as I said, we know with building-related illness that there are some other conditions like Legionnaires. There are several occupational diseases, sometimes grouped together as inhalation fevers. And then we have this whole mystery area of the unknowns. The quote, sick building syndrome. And there are lots of people who don't like this term. There are lots of people who think this is not a real condition. And there are lots of people who think that if it is a real condition, it's associated with so-called toxic black mold. And there was a cluster in Cleveland quite a while ago, over 25 years ago, of children who had a hemorrhage condition. They found this black fungus in their house. They didn't take pictures. And the fungus was identified as something called Stachybotrys, called Stacky in the trade. And this fungus is known to make some terrible, terrible fungal toxins called trichothecenes. And it's kind of like ever since there's this notion of toxic mold is out there, and if you get it, this black mold, and hey, there are dozens of common species of mold that is black, that is not Stachybotrys. People will see something black growing in their house, and they think, I have got this horrible, horrible toxin, and it's causing all these symptoms that are associated with sick building syndrome. Now, when you look at those symptoms, you see lots of us have insomnia. We get headaches, we get dry, itchy skin, nothing necessarily to do with fungus, and so it's a suspect diagram. And I'd like to say that it's almost like it's a legal concept, not a mold concept, and that this so-called toxic mold disease is not accepted by the medical community. And but people who believe in it think it's caused by mold. And I would say once every 3 weeks I get a call from someone who's found me wanting to know about their mold disease. And there are many companies out there that they're being entrepreneurial right in this moment. And some of the ads they put out, like the National Treatment Centers for Environmental Disease is not a federal agency. It's a profit-making entity, and I think that it's unfortunate that, because I believe that molds can make you sick, that so much of what's happening out there is pure shysterism. So, to my own story, I spent almost my entire professional career at Tulane University, which is located in New Orleans. And during the years there, I was a specialist in mycotoxins. And because I'm very skeptical about mycotoxins being the cause of this so-called sick building syndrome, I actually sent one of my children to a very expensive college by regularly taking consulting assignments for insurance companies and saying, hey, I don't think the mycotoxins did it. And for that reason, I had a lot of information about sick building syndrome in my brain, but that's not what I worked on. I did the genetics of mycotoxins, and the fungus I had studied all my life was about had just been sequenced, and I had a sabbatical, and I was going to go and learn how to annotate it and have a wonderful time doing fungal annotation for the rest of my life. But 2 weeks before I was going to leave on sabbatical, there was a hurricane. Here's a picture of New Orleans. You see there's a huge lake to the north of the city. That windy thing is the Mississippi River. And much of the city is below sea level. And I love this picture. A great big lake north of the city, New Orleans. The levees broke, several of them. And this is what happened. I happen to love New Orleans. I can't say I love New Jersey, but this is motivation for maybe moving to higher ground. And actually, after the hurricane, when people were tracking me down, I used to send out emails that said, senior scientist seeks position above sea level university. Tulane Medical School. This is my friend from Loyola. My husband and I had evacuated. He's from New Jersey. He's a Rutgers grad. We came to New Jersey. I didn't get home until early October, so I called a friend at Rutgers and asked if I could make media because I knew a lot about that just add water. And I'm going to just show you some pictures of my own home. The water had come in and knocked furniture over. I've had the clock restored, but everything else got thrown away. Something that the pictures don't show, it smelled horrible. I actually had a conversion experience, and ever since then I've been studying the aroma compounds that molds make. And I am more of a believer in sick building syndrome than I was before. I don't think that mycotoxins are never involved, but I don't think they can be the only thing. Relatively little work was done at that time. I think partly because most of us who were there who could do the work were completely crippled. No autoclaves, no electricity, no nothing. And a lot of the people who came in were looking for the wrong things. They're looking for cholera and heavy metals, and surprisingly little was done on the fungi. There has been some post-Katrina research on health, and this paper published in 2010 said, hey, no correlation. I find it hard to believe that there are no health effects, but partly it's just like when they first studied after the BP oil spill and said no effect on the bacteria. It depends on what you're looking. So in my lab at Rutgers, we have been studying the volatiles. I'm not going to go there. The most exciting thing we found is that one of the most common, we can give Drosophila flies Parkinson's disease in a model, and we've done all kinds of neat molecular biology, and I can't get NIH to give me money to study this, which I think is really a shame because I think that we are ignoring that part of the microbiome. That's a whole other talk. So I'm going to end with what you want to hear. And as I said, I had thought I'd be following lots of talks which would tell you about the wonders of high-throughput sequencing and how fast and how cheaply we can now come in and monitor fungal environments. I'm going to repeat what I said at the beginning. When you're doing the microbiome, it's even more complicated than perhaps the human gut because there is such a variation in what we're studying. Where is your building? Is it in Stockholm? Is it in Saudi Arabia? Is it in New Orleans? Is it in New Jersey? I mean, the parameters can boggle your mind. But we've been asking basic questions like, what's there? We try to find out how it got there. And then, as I said, we try to figure out where interactions are. As you might expect, the first microbiome studies have been done in places that we're interested in, schools, a lot of sampling in bathrooms. Microbiologists love to sample bathrooms. Homes. This is one big study where they went around 9 common places, and basically you find out that you have different populations where there's a lot of food, different places, populations where it's a lot of dust, and then what we touch has yet a different population, and that if you go into different houses, you'll find that those different places tend to be more the same than within a given house, meaning if there's food there, you're going to have the same kind of, more or less, populations of microbes. The way in which a building is ventilated, how well it's kept, all of these factors, whether you have a pet, all have big impacts on the microbiomes that are found in houses. And as a person who works with fungi, I kind of have a hang-up about this. Most of the indoor microbiome studies have used bacterial probes, and so they only describe the bacterial populations. But when you do that, you find that a huge component of the bacteria that you pick up inside comes from us. And we leave remarkable signatures. I think someday they're going to be doing forensic stuff on criminals just by what they have shed off. And so if you have a healthy person in there, it's a very different thing from having a sick person because they're shedding what they carry. And this slide just says what we find inside is a reflection of what we carry inside. We build these buildings so we can occupy them, and when we occupy them, I think we've greatly underestimated how much of an impact we leave on them. And so this slide talks about the work that I think we're going to be hearing a lot about in the next couple of hours. The human impact on spaces is enormous, and we'll be picking up, depending on the space and all these other variables, a human microbial imprint. It will vary depending on how long the people have been there, what they're using the building for, and of course there will be variables that have to do with cleanliness, ventilation, et cetera, et cetera. Whenever I read these indoor microbiome studies, I'm reminded of this character. I think that Charles Schultz was prescient. Do you remember Pigpen and this person going around shedding? Well, apparently we all do this. We do it all the time. We're shedding thousands and thousands of skin cells that carry our microbiomes and leaving them in our spaces. And this little cartoon figure should be the mascot of indoor microbiome research. There's been a big study done at the University of Chicago on hospital microbiomes. They actually are a wonderful place to do the study because you can control stuff better than you can in a school or in a shopping mall or in the Hoboken station. There's been a lot of publications, and I have to say, in a way, it's disappointing. When we use the culture-independent methods, we find out that, hey, there's more microbial diversity than we thought there was, but we haven't necessarily found anything that just lights up and says, oh, that's what's making us sick, or that's not what's making us sick. We found that the fungal populations tend to come from outside, the bacterial populations tend to come from us and our dogs and cats, and the just add water factor, how wet it is, whether it's a humid climate, whether there's condensation behind the walls, et cetera. I've been surprised how frequently I'm asked this question, hey, now that we can study microbiomes, can we make them healthy and keep our indoor environments right? The whole notion of, yogurt for the building. Well, hey, we don't even understand what's making us sick, so it's pretty hard to predict that we're going to be able to create healthy microbiomes. But I would imagine there's going to be an entrepreneur out there, not necessarily an ethical one, who does this. There are a lot of problems in the ways in which we study health risks and the presence of indoor fungi and other microbes. And much of what we do is a one-on-one association where we don't have the brain power or the thinking power, the synthesis power to develop the right algorithms for the risk assessment. We are looking at combined risks, and we are not good at that. We're getting better, but we're not good at it. So, repeating what I've said, the kind of upkeep of your environment makes a difference. It will infect what you find there. The life forms that are in our buildings will affect the building. And the use of the building will affect what microbiomes we find there. Every single one of us who gets involved in microbiome research has this feeling. There's too much data. And I like this picture because as I have been trying to corral building scientists, chemists, pulmonologists, architects, and microbiologists to write a report about, quote, microbiome of the built environment, we're drowning in data, and we're not drowning in synthesis. And with some thanks, this is a couple of pictures of my new lab at Rutgers. Rutgers not located in a city with as good food as Tulane, but the science is quite good. I want to especially thank Jason Richardson, who has helped me with the Parkinson's work. I've had some interesting scientists come from, especially the Middle East. We found some of our volatiles make plants grow better, and there's a lot of interest in that. Jack Gilbert at the University of Chicago has been one of the big spokesmen doing the bacterial microbiome and is a wonderful cheerleader for the field. And finally, the Sloan Foundation, which has funded some of the work in my lab and which is the main funder for work on the indoor microbiome. NASA puts a little in. NASA doesn't have a normal indoor microbiome because they can't ventilate, but they've got a big problem. And so with that, I will stop, and I'm available for questions. Yes. He wants to know if there are fecal transplant opportunities, and nobody likes thinking about it. It's amazing how much fecal imprint there is in indoor environments that have nothing to do with restrooms.
My question, though, or questions are, to me, thank you. To me, it would seem that the environment you're looking at, or the ecosystem, is even more complex than the human microbiome environment and ecosystem, because it encapsulates all of the human condition, plus many different types of buildings and environments within buildings, and then buildings and cultures and environments around the world that are very, very different. So
that's right.
I don't know if that's a good quick view to take on the field that you're describing. The other question that I had was, is there a science and research developing on the building side, like what the microbiome and the human microbiome trafficking through buildings has on the way that buildings stand up and function, and research opportunities for translational science on the architecture and engineering side of buildings?
Right. Well, there's huge interest in from the building side, and one of the charges of this National Academy study is to try to at least figure out what's going on. My sense is it's way too soon, and what we have figured out in the past is not just to be thrown away. Ventilation's important, and we're finding that there is actually a kind of a battle going on between energy efficiency, and I didn't talk about that, but a tight building that you don't change the air in very frequently is associated with some of these mysterious sick building syndrome things, and probably with changes in microbes and the chemistry of the air, but we don't know. And so balancing the need for energy conservation with a healthy microbiome is a problem, and it's way too soon to start giving recommendations. And I get frustrated when people ask for this, quote, yogurt for the building. We don't even know what is making most people sick with these vague symptoms, so how are we going to fix it, you know?
So what do you see as the outcome of this study? And what kind of information would you like to see made available to consumers?
Well, I think one big outcome of the National Academy study is going to be to point directions where funding agencies should be putting some money. It's very scattershot right now, and like a lot of other microbiome research, it's not necessarily the result of a strategic plan. You know, so we're trying to create somewhat of a strategic plan. I think with the second part of your question, I'd like to quote a book that was popular when I was young and raising my children. It was Dr. Spock said, "You know more than you think you do." We have been studying indoor environments for a very long time, partly to keep them from rotting, but partly also because there was a time when people thought, for example, that bad air caused illness, or that the clothing associated with sick people caused illness. And there's a great paper that Brent Stevens from Illinois Institute of Technology published just a couple months ago, where he actually gets a paper from some British study done in the 1850s and compares it with some of the modern microbiome research. And you know, they weren't that far apart. So I think we already know a lot about what's good for buildings. We're adding a layer of complexity. And hey, we may make some new breakthroughs, but right now we don't have a lot of breakthroughs.
How do you think the changes in the piping to buildings? There used to be a lot of copper piping, which apparently has some bacteriostatic properties, but the changes in the PVC pipes and this sort of thing, how do you think that changes?
Boy, you know, that whole thing about plumbing is another area, and I didn't even go there in my talk today. Our study has brought in several speakers who specialize in water, and yes, there are all kinds of issues with the water supplies, and I emphasize that when you get the water into the building, that's often when the microbial populations start doing interesting things. Remember, with microbiome research, when you're just doing the nucleic acids, you don't know whether it's dead or alive or if it's going to grow. You're getting a snapshot of what's there. When you add the water, that's when things start to grow. And yeah, I don't know. And you'd need a plumbing expert on that one, because it's, as I say, it's most of what I have been focusing on has to do with surfaces and walls, floors. Yeah, one.
Okay, very interesting talk. My question is, do you, because as you talk, this building environment will affect the human health. So by the microbiome in this building. So do you know, or is other publication shows how long, like this share or commute this bio in the building with the human, this bio in the human bodies? Or my key point is if that take longer times, we can just transplant or plant the health microbiome in the building instead of taking some medicine for human, just make our health if are sick.
I'm sorry, I don't quite understand your question. Are you saying that sick people will make a sick microbiome?
Yes. I mean, so how long do you know? How long take this time for share this this organism?
Oh, you mean sharing? Oh,
Yeah.
okay. Well, there too, century of microbial research, and we know a lot about transmission of flu and measles and viral diseases, and so it depends on which microbe, and that is all under the category of infectious diseases we understand. I chose in this lecture to emphasize the ones we don't understand, And so you can't make a generality about how long, because it depends on whether we're looking at transmission of Legionnaires or of influenza or of other things that people might be sharing. I think you're sort of asking a huge ID, infectious disease question, or maybe I misunderstood it. If somebody else had a sense of what the question is. I'm not sure I got it, but anyway, that's why. Well, thank you very much.